Now it should be good. Okay, oops, what happened? I just updated that schedule. Where is it? Well, look at the early class. It's the same schedule, but I know that one got updated. So open that one. I just added some lab dates on there. And I had started putting together a lab for this week. That would be due next Wednesday. But then I changed my mind. Yeah, so the lab number 9 is due tonight, mitosis and meiosis. Everybody, how many of you haven't done that yet? Be honest. Need another day? I'll give you another day. Okay.
Thank you.
But so we have an exam next week. So I'm not going to give you a lab. Just spend your time studying. But the lab I was going to have this week is a sheep brain dissection. You got a sheep brain in one of your lab kits? Is a little sheep brain. So that'll be our next lab activity, just dissecting that sheep brain and learning the parts of it. Sheep brain looks just like your brain, except your brain might be a little bigger. It's getting bigger all the time. Every week, the more anatomy you learn, the larger your brain gets. And I was planning anyway to have a total of 12 labs, but we'll see. You got an eyeball, I think it's a cow eye, also in one of your kits, so we might do that. Maybe cut that open, see what's inside. What else? Okay, so next week's exam, the schedule says chapters 10, 11, and 12, but somebody pointed out to me today that on Moodle there was a notice that somewhere that lecture exam number 4 would be on the 11th, but it had the chapters, I think, only 10 and 11. But I wanted to include 12 in that, chapter 12, which we're going to cover today, because otherwise we have only one exam after that, and if we don't include chapter 12 in next week's exam, then the last exam is going to cover 4 chapters. I'd rather have 3 and 3 than 2 and 4. So I'm planning to have chapter 12 on next week's exam. Just get that chapter out of the way. Okay, and I'll be here early. I'll be here at 5 next week if anybody wants to come for a review session. Okay, anything else you need to talk about? Presentations in 2 weeks, right? Everybody at least have a topic in mind. If you don't have one, I'll help you come up with one. And if you're not sure if your topic is any good, ask me and I'll tell you what I think of it. Okay, so we finished the muscular system, chapters 10 and 11. Now we're entering the last organ system that we're going to cover in this course, which is the nervous system. And there's 4 chapters on that organ system. And we're going to do chapter 12 today, which most of this chapter, well, first of all, it's divided into 2 parts, part 1 and 2. And that wasn't me who did that. I had made the PowerPoint for this chapter based on the old book a few years ago, and Charlotte Carter, who also teaches this, she has updated some of these chapters to match the new textbook that you're using better. And so when she did that for this chapter, she split it into part 1 and 2. I don't know why. Maybe just because it's so long, but I went ahead and used it that way because I didn't have time to do otherwise. And sorry I didn't get these on there until today, these PowerPoints. As usual, I'm barely keeping up. Oh, I already had that open. So part 1. Got to redo this. It's recording, right? Okay, nervous system. Okay, nervous system and the endocrine system are 2 systems of communication within the body. So the nervous system is how different parts of the body communicate, mostly the brain communicating with everything else. How do I get back where I was? But the nervous system communicates with nerve impulses along neurons, and the endocrine system uses hormones. Nervous system is much faster than the endocrine system because nerve impulses are just lightning fast. They go from your brain to some other part of your body, or from some part of your body to your brain in a tiny fraction of a second. Neurology, of course, the medical specialty of nervous system. You need to know this organization, or how the nervous system is can be divided up into subdivisions. First of all, there's a central nervous system, which is just the brain and the spinal cord, and then the peripheral nervous system is everything else. Peripheral nervous system includes the cranial nerves and spinal nerves. And look at this diagram here that shows the cranial and spinal nerves. First of all, the word nerve, we need to define that word. A nerve is not the same as a neuron. A neuron is a nerve cell, but a nerve is a bundle of the axons. So here's a neuron, a nerve cell. So this long extension is the axon of that neuron, and a nerve is a bundle of those axons of many different neurons. So the cranial nerves and spinal nerves are bundles of axons of neurons. The cranial ones connect directly to the brain. A cranial nerve is one of these bundles of axons that are connected directly to the brain, and then they extend out into various parts of the head and upper body. And the spinal nerves are the ones that are connected to the spinal cord. So they're not, spinal nerves are not part of the spinal cord. They're not in the spinal cord. They are these nerves here that are connected to the spinal cord. And they're in pairs, left and right, all the way up the spinal cord. So the cranial nerves and the spinal nerves connect the brain and spinal cord to various parts of the body. And they make up the peripheral nervous system. Then the peripheral nervous system can be divided up further into the somatic, the autonomic, and the enteric nervous system. Somatic means conscious and voluntary, and we saw that word already when I was describing the neurons that control the skeletal muscles. Those are called somatic motor neurons. Somatic means conscious and voluntary. So we always have information coming into our brain and going out of our brain. If the information coming into our brain is conscious, that just means that we're aware of it. You're aware of that information coming in. Like if I'm talking to you, your ears are hearing that sound and then sending nerve impulses to your brain. That's conscious information. You know that information is coming in. If you're wearing itchy socks right now, that information is coming into your brain about that. But there's also information coming into your brain all the time that you're not aware of. For example, we have sensors or sensory receptors in some of our blood vessels, the aorta and the carotid arteries, that are always measuring your blood pressure, sending information about your blood pressure to your brain. You don't even know that's happening, right? So that's unconscious information. The somatic nervous system is made up of the neurons that have conscious information coming into your brain and voluntary information going out. That means when you consciously decide to do something with some part of your body, then your brain will send out that information to that body part. That would be also somatic nervous system. And there's only one kind of organ that you can voluntarily control. What is that? One kind of tissue or organ. We just did 2 chapters on it. The skeletal muscle, right? There's really not anything else you can consciously control. Even when I'm talking, I'm consciously controlling that, but it's all muscles that are doing the speaking. If you can breathe, you can decide to breathe faster or slow down your breathing. But again, that's not your lungs that you're controlling, that's your breathing muscles that you're controlling. So everything except your skeletal muscles is involuntarily controlled. The autonomic nervous system, that's unconscious, involuntary. So any information coming into your brain that you're not aware of it, that's autonomic nervous system. And any involuntary control of any organs, that would be autonomic. So, for example, again, you have these sensory receptors in your blood, some blood vessels sending information to your brain about blood pressure. If your blood pressure starts to drop a little bit, your brain might decide to send information to your heart to make your heart rate speed up a little bit. That would be involuntary control of your heart. So that would be through the autonomic nervous system. Autonomic, or the need to define the words sensory and motor. Sensory is just information coming into your central nervous system, your brain and spinal cord. Motor information is information going out of your brain or spinal cord to your organs to control that. The motor part of the autonomic nervous system, that is the involuntary control of your organs, can be divided into sympathetic division and parasympathetic division. Sympathetic division, when the sympathetic division is controlling your organs, that means that we say that you're in the fight or flight condition. That means there is some kind of emergency, life or death situation, and so fight or flight means that you have to either fight for your life or you flee, you run away. You can't actually fly, you don't have wings. Flight means to flee. So you're either going to stand and fight or you're going to run for your life. And that's so when that happens, certain organs change, so your heart rate speeds up, your airways open up so you can suck in more air, you get more oxygen to your skeletal muscles, and the blood vessels leading to your skeletal muscles dilate to deliver more blood, more oxygen, more nutrients to your skeletal muscles, because you might have to be running from a grizzly bear or something. And also your adrenal glands will release the hormone adrenaline. That also has a similar effects on your organs. It speeds up your heart rate. Meanwhile, your digestive system shuts down when you're in the fight or flight situation. It's not important that you're digesting your lunch if you're running from a grizzly bear. So the rest of the time, when you're not in the fight or flight situation, then you're in the rest and digest position, which is this means you're not in a life or death situation. Your digestive system is functioning; it's not shut down, and that's the parasympathetic division of your nervous system. So you have those organs that like the heart and airways and some blood vessels, all those organs that are affected by the fight or flight condition, they have two sets of neurons leading to them. They have a set for fight or flight and another set for rest and digest. So most of the time, the parasympathetic neurons are controlling your organs, but something happens. If somebody walks in that door with a machine gun and says, "I'm going to kill all of you," we're all going to go into fight or flight position immediately. When that happens, for my online version of this class, I have an essay question about this. Why is it that, for example, if you're driving your car and a dog runs out in front of your car, and you don't want to kill a dog, you don't want to run over that dog, so you you go into fight or flight. Your heart speeds up, you hit the brakes, and you almost swerve into another car and hit it, and so now your heart's pumping really hard, but the dog runs away, and the dog's just fine. You didn't hit any other cars or anything. The emergency's over. Why is it that for a few more minutes your heart's pounding? Your heart doesn't slow back down right away, even though the emergency's over. Why is that?
The signal has to be sent all the way to the body that everything's okay.
Well, but the nerve impulses happen lightning fast. It shouldn't take long for your nervous system to go back to normal. Exactly, it's the hormones. Because when you go into fight or flight, your brain sending nerve impulses to your heart and other organs to speed up, and also sends nerve impulses to your adrenal glands that release the adrenaline into your bloodstream. And now that hormones in your blood, that even when the emergency is over, those hormones don't disappear immediately. Those hormones are going to be there for a while, and that's why your heart keeps beating for a few minutes. It takes a while for those hormones to be cleared out of your blood. Hey, oh, and finally, the enteric is the third division of the peripheral nervous system. That's for, it says, regulating the GI tract. That GI stands for gastrointestinal. That's your digestive system. So your digestive system has its own nervous system. And that's also involuntary and unconscious. You can't voluntarily control your digestion. It just happens automatically. Hey, okay, just more information about that. Central nervous system, brain and spinal cord. The word integration. Integration just means to combine stuff together. And so that's what happens in your brain. Your brain is also always receiving a lot of different kinds of sensory information. Stuff that you're hearing and seeing and smelling and tasting, and also the unconscious information that's coming in from all these sensory receptors in your body that are measuring your blood pressure and the amount of oxygen in your blood and your blood sugar level and your body temperature, sending information to your brain about all that stuff. Your brain combines all that information. That's called integration. And then your brain decides what to do about all that information. Whether it should respond somehow, change some physiological functions, speed up your heart rate, cause you to breathe faster and deeper, release some hormones, or store some memories in your brain for later. Peripheral nervous system, I already talked about that. Somatic, autonomic, enteric. Talked about that. Sympathetic, parasympathetic. We'll define that word ganglia and the plexus later. Diagram, like a flow chart with the same information about the organization of the nervous system. Those arrows show the direction of information. So sensory information coming in, motor information going out. Again, these words sensory, integrative, and motor. Sensory information is information coming into the central nervous system. Motor information is going out to control organs. and the integrative is in between. It's in the brain and spinal cord. So the neurons that take information into your central nervous system are called sensory neurons. Motor neurons take information out, and the ones in between, which are in your brain or spinal cord, are called interneurons. Inter means between, and those are the ones that do the integration. That's where most of our neurons are. You have over 100 billion neurons just in your brain, processing all this information that's always coming in and going and deciding what to do with it, what information to send out. We already learned this back in chapter 4, I think, when we studied tissues. Nervous tissue includes two types of cells, the neurons and the neuroglia. The neurons are the actual nerve cells that conduct nerve impulses. They're the ones that do the communicating. But there's also neuroglia, 6 different kinds of neuroglia that are helpers for the neurons. And we'll see a little later what those 6 types of neuroglia do. Okay, most of the time that we're going to spend tonight is going to be learning about how nerve impulses happen. And the word action potential means the same thing as a nerve impulse. And so you gotta, before we can learn how an action potential works, we have to explain about another kind of nerve impulse called a graded potential. And to learn about that, we have to learn about gated ion channels and membrane voltages, and we have to review some stuff that we learned before. So it's just it takes a long time to put all this information together before we can learn what's actually happening in a nerve impulse. It's not really that complicated once we finally get to it, but it's why I don't like teaching. It's just a lot to explain. And people go to sleep because it's not very fascinating, not very interesting. At 7 o'clock at night after a long day. Okay, it says here that neurons have electrical excitability, and a nerve impulse, it conducts along the axon of a neuron, and it's sometimes described as being similar to electrical current flowing through a metal wire, but it's really not that simple. There's a lot more to it than that. Let's look at more detail about the structure of a neuron before we go any further. So that's the diagram of a neuron. The big wide part at one end is called the cell body. That's where all the important organelles like the nucleus and the ribosomes and mitochondria are located. And those cell body typically has a bunch of those branched extensions sticking out. Those are called dendrites. Word at the very top, dendrite. The word dendra is it's Latin. It means tree. So those are like branches of a tree. And you see the arrows along those dendrites. That's indicating that there are impulses on that those dendrites receive, and then they conduct those impulses into the cell body. And then there's that one very long extension called the axon, and that's where the actual action potentials or the nerve impulses go from the cell body all the way along that axon to the ends, the other end, and so you see the arrows that nerve impulse starts there where the axon is connected to the cell body. That's called the axon hillock. It's labeled there, hillock. And then that nerve impulse conducts all the way along the axon. This axon can be 3 feet long. It can be, go all the way down your leg. One neuron, one axon. It's one long cell. And at the end of the axon, it's usually branched. So this, whatever nerve impulse comes along here, that nerve impulse will go out all of these branches. And these axon terminals at the ends, the word terminal just means end. Each of those axon terminals will connect to something else, either a muscle fiber there, stimulating a nerve fiber to contract, or it could they could connect to some other organ or a gland, telling a gland to release a hormone or something, or it could connect to the dendrites of another axon, and cause the dendrites of another neuron. And then it could cause that second neuron to send out an action potential or nerve impulse along its axon. The word fiber is another word for axon. So again, we use that word fiber for a lot of different things in anatomy. If we're talking about neurons, the word fiber means the same as an axon. And these, I didn't mention these things that look like hot dogs or sausages. It's shown here. That's a bunch of layers that are wrapped around the axon. That stuff is called a myelin, the word myelin. Each one of these is called a myelin sheath. The word sheath just means a cover or something. And that myelin is a lipid, fatty material that is like insulation on the axon. It's kind of like the plastic insulation on a metal wire that it insulates, covers and protects the axon, and it also speeds up the action potentials along the axon, because when an action potential is coming along the axon, instead of conducting all the way along the axon, it jumps over the myelin sheaths, and that speeds it up. There's a disease called multiple sclerosis. We know about that. Might nobody know someone who has it. Not real uncommon. That's believed to be caused by the person's own immune system attacking the myelin sheaths on the axons of the neurons. And well, it can cause if there's no longer that myelin, then the action potential will slow down. So they lose coordination. And also, just like if there's two electric wires touching each other and the insulation is missing, they're touching, so current is going through one, it'll short circuit into the other one where it's not supposed to be. The same thing can happen if there's two axons side by side and the myelin's missing. A nerve impulse could be coming along one and it could jump onto the other. Now a nerve impulse is going to the wrong place. So that's why people with multiple sclerosis, they can be just fine, and then all of a sudden the leg will go numb. They'll lose control of the one leg. And I had a student about 8 years ago. The job I had the year before I came here, it was a class just like this, and I was talking about a condition called sciatica, which is it's caused by a nerve pinched in the lower back, and a nerve that runs down the leg, and it causes pain and numbness. I had it once myself for a few months once, and it feels like something's poking you real hard right in the butt, right in that knot between your hip and the gluteus maximus muscle. That pain can run all the way down the leg, all the way to the ankle sometimes. But I was talking about that, and there was a student in the class, and she said, "Did that cause your whole leg to go numb?" And I said, "I never heard of that, but I guess maybe." And about 3 weeks later, she was diagnosed. She asked that question because that happened to her. It turned out she had MS. She was about 35 years old, and she wanted to be a nurse. She was all alone. She was single. Now she had MS, and her life was ruined. It was really sad. I don't know whatever happened to her. Okay. Neurons can have different forms. Most of them are this kind, like the diagram we saw before. It's a cell body with dendrites sticking out and one long axon with branches. But some of them are like this. Got a cell body and an axon, but one dendrite with a bunch of branches, and then some of them are like this. It's like an axon with the cell body sticking out the side of the axon, and then the dendrites at the other end. And they have names: multipolar, bipolar, unipolar. But I'm not going to ask those words on an exam, so don't worry about those. A synapse is just where the axon terminal of one neuron meets something else. If it's a muscle fiber, then that would be a neuromuscular junction, and there would be a synapse there. It could be the axon terminal of one, well, like shown here. This is an axon of that neuron. These branches all have axon terminals. In this case, axon terminals are forming synapses or connections to the dendrites of this other neuron, and also this neuron. In this case, these axons of those neurons are connected right to the cell body of this other neuron. Or this, like I said, these axon terminals could be connecting to a muscle fiber or some other kind of organ that is being controlled by those neurons. So a synapse is just where the neurotransmitters are released from an axon terminal that's connecting to just some other kind of cell. Again, we already saw these words: sensory, motor, interneurons, or association neurons. These words afferent and efferent. Afferent just means something that's flowing in to something, and efferent means flowing out. In this case, it's nerve impulses. So, so sensory neurons have information flowing into the brain, and motor neurons have information flowing out. We'll see those words again, and some blood vessels are described as afferent or efferent. Like in the kidneys, there's blood vessels taking blood into the kidneys, and those are called afferent arterioles, and there's blood vessels taking blood out of the kidneys called efferent arterioles. Sensory receptors The sensory receptor is just any of many different kinds of structures that we have in our body that are measuring something, measuring or monitoring something, and then sending information to the brain. So there's a few examples here. These tactile epithelial cells. So these are what we have in our skin. Cells that are sensitive to very light pressure, and so any pressure on these cells, it will cause a nerve impulse to be sent through this neuron to the brain. So, even though it seems like what we're feeling is at the source of that pressure, it's actually in your brain where the sensation happens. So in some cases, like these other kinds of touch or pressure receptors in the skin, the receptor is actually part of the neuron. It's not separate from the neuron. It's just the modified dendrites, so that any pressure on that corpuscle will cause a nerve impulse to be sent to the brain. These nociceptors, nociception means pain. These are pain receptors, and they're just bare dendrites. These are what are sometimes referred to as nerve endings. You've probably heard of nerve endings. So they're just dendrites of neurons that, if there's any kind of disturbance or damage to these dendrites, it'll send nerve impulses to your brain, and you'll feel that as pain. Okay. Neuroglia, these helper cells, are sometimes called glia or just glial cells. There's way more of these than there are neurons in the nervous system, so they're important. And there's 6 kinds here. The first 4 listed here are in the central nervous system, and the other 2 are in the peripheral nervous system. So we need to talk about each of them just a little bit. These astrocytes, astro as in astronomy, so site means cell. So these are shaped like little stars, and their function is to hold neurons in place, and also in the brain they form the so-called blood-brain barrier. In the brain, unlike most other organs in your body that have a lot of blood capillaries running right through the tissues to deliver oxygen and nutrients, in the brain you don't really have a lot of blood vessels running deep into the tissue of the brain. Instead, there's a blood-brain barrier, and your brain has a fluid, a liquid circulating through these hollow spaces inside your brain and around the surface of the brain, and also down through the spinal cord and back up. This liquid is called cerebrospinal fluid, and that cerebrospinal fluid is what delivers oxygen and nutrients to the blood cells, or the brain cells, and carries the waste products away from those brain cells. But there's a barrier between the blood and the cerebrospinal fluid, and that's called the blood-brain barrier. And that barrier is formed by these astrocytes. One of the I guess functions of that blood-brain barrier is that it prevents any infectious pathogens that might be in your blood from getting into your brain because of that barrier. And so that's why you don't, infections of the brain are not very common. Although there are, that's, meningitis is a infection of the brain. It can be bacterial or viral. Even fungus can cause that. But that's on the surface of the brain. It's not in the deep tissues of the brain. One of the problems caused by this blood-brain barrier in medicine is it prevents some drugs from getting into the brain tissues, especially in the case of brain cancer. That causes a challenge for doing chemotherapy for brain cancer, because the chemotherapy drugs cannot get past the blood-brain barrier into the brain where the tumors are. So it just creates a new challenge for chemotherapy for brain cancer. Hey, oligodendrocytes, these make the myelin sheaths that cover the axons of some neurons. So here's an oligodendrocyte cell, and it sends out these extensions that produce the myelin that wraps around the axon to make those sheaths. And this is only in the central nervous system. In the peripheral nervous system, the myelin is produced by another kind of neuroglia cell called the Schwann cells. Microglial cells, these are actually the white blood cells of the immune system that live in the nervous system tissue, nervous tissue. So these are macrophages, which are just large phagocytic white blood cells that eat bacteria and viruses if they find them. Ependymal cells, these are cells that produce the cerebrospinal fluid that's in the brain and spinal cord. And so these hollow spaces that contain that fluid are lined with these ependymal cells that produce the fluid, and also they have cilia that squeeze the fluid to keep it circulating. Here's a diagram showing the different kinds of neuroglia of the central nervous system. And now in the peripheral nervous system, there's the Schwann cells. These are like the oligodendrocytes of the central nervous system, and they make the myelin sheaths around the axons. And these are also important. They also function in regenerating injured axons. So if you have some kind of injury, a cut or something that cuts through some neurons, and now these neurons are not connected. They're not connecting your hand to your brain anymore. So you might have numbness in part of your hand, or you might not be able to control some muscles of your hand. But that feeling and movement might come back after several weeks or a couple of months, because those neurons can regenerate. And the Schwann cells function in that regeneration of injured axons. Schwann cells are only in the peripheral nervous system. We don't have them in the central nervous system of the brain and spinal cord. So if those neurons get severed or damaged, they typically don't regenerate. And finally, the satellite cells, they also hold neurons in place. It says regulate exchange of material between neuronal cell bodies and interstitial fluid. Interstitial fluid is just the tissue fluid surrounding cells in any tissue. The fluid in between the cells. And some of these neurons they're so specialized for doing only one thing, which is conducting nerve impulses, that they're pretty helpless. They can't even function by themselves to do the normal things that other cells do, like absorbing nutrients to keep them alive. So they have to have helper cells to do that for them. Okay, a little more about these myelin sheaths. The nodes of Ranvier, these are just the gaps in between the myelin sheaths. I'll back here. So each of these gaps is called a node of Ranvier. I guess that's the name of the guy who discovered them or something. Yeah, so myelination is not complete by birth. So in a child, the axons are not fully myelinated yet, so the nerve impulses are still slow, and that's why they're not as coordinated as adults in some of their movements. And not all axons have myelin. Some axons are myelinated and some are unmyelinated. So we never do have myelin on them. Okay, this table just listing again, summarizing the functions of these different neuroglia. Some other terms. The term ganglion and nucleus mean the same thing, except in a peripheral nervous system versus a central nervous system. But it's just a cluster of cell bodies, neuron cell bodies. And there's that word nerve, a bundle of axons in the peripheral nervous system. So the cranial nerves and the spinal nerves are bundles of axons. But the word tract means the same thing, except if it's in the central nervous system, in the spine, usually. So a tract would be a bundle of axons running up or down within the spinal cord. Okay, white matter and gray matter. Everybody's heard of this. The brain is made up of white or gray matter, but what does that really mean? What is the white stuff in white matter? It's myelin. The myelin is white. And, so the white matter is bundles of myelinated axons, and the gray matter is everything else that does not have myelin. So cell bodies, dendrites, bundles of unmyelinated axons, and neuroglia. And in the brain, you can see the gray matter is on the outside and white matter is on the inside. This outer part is of the cerebral cortex. The cerebrum is the largest part of the brain. That is the cerebrum. It includes the outer part. The very outer part out here is called the cerebral cortex. The word cortex just means the outer part of an organ, and this is mostly cell bodies of neurons out here, and the axons, the myelinated axons from these cell bodies out here extend through the deeper parts of the brain, leading to the spinal cord, and so that's why why the white matter is on the inside. But the information from these cell bodies out here is coming to or from the spinal cord through this white myelinated axons. In the spinal cord, it's just the opposite. The gray matter is on the inside, and the white matter, myelinated axons, is on the outside. Yeah. Here we see the brain again, and so out in the cortex, some cell bodies, and then the axons would take the information out of the brain, down the spinal cord. And so here would be here's the gray matter, and then myelinated axons leading out of the spinal cord into a spinal nerve. This would all be motor information going out of the brain to the muscles, and meanwhile, sensory receptors send sensory information through the sensory neurons into the spinal cord, then up to the brain. Okay, so leading up to the description of an action potential, we have to talk about ion channels. Remember we learned about diffusion, simple diffusion, and facilitated diffusion. And I have said in here before that once you learn something, you can't forget it because it's going to come back. Now we got to use that information. Remember, simple diffusion is how some substance can get into or out of a cell if it can pass right through the phospholipid bilayer of the plasma membrane. So that's how oxygen comes in to a cell. Carbon dioxide comes out of a cell. Water can pass through. But there are other substances like ions that cannot penetrate the phospholipid bilayer, but they can come through some channel proteins in the plasma membrane. The cell just has to allow those channels to be open so the stuff can diffuse in or out. That's called facilitated diffusion. That's what these ion channels are about. Just channel proteins in the plasma membrane of the neurons that allow ions to flow in or out of the neuron. And the first one is just called leak channels, and they just open or close for no reason, just at random, to either allow or not allow ions to come in or out. But these other three, they're called gated channels. That means they have like gates or doors on them that open or close, either open and allow ions to come through or close and not allow them to come through. And they open or close for different reasons, depending on what type of channels they are. Ligand gated, these should be called chemical gated or molecule gated because they only open if a certain kind of molecule comes into contact with a receptor on that channel protein. Normally they're closed, but if something like a particular neurotransmitter molecule touches it, it'll cause the gate to open. Now ions can flow in or out. Mechanical gated, some kind of pressure would cause these ion channels to open. So that's how the touch receptors, some of the touch receptors in your skin work. There's just any pressure on on your fingertip will cause a deflection or a stretching in the plasma membrane on these neurons in there. It will cause these gates to open, and now ions rush in or out and cause an electrical change that will lead to a nerve impulse going to your brain, telling your brain that there's something putting pressure on that part of your body. Then there's the voltage-gated channels. The change in the voltage, the electrical voltage at that area on the plasma membrane of that neuron will cause the gates to open, allow ions to come in and out. So that's so here's the leak channels. So there's different kinds of these for different kinds of ions. So there's potassium K+ leak channels and sodium Na+ leak channels, and they just open and close randomly. So there's always potassium ions and sodium ions leaking in or out of cells through these leak channels. The ligand-gated ones are chemically gated, so normally it's closed, but this would be for potassium. So this one is for potassium, and calcium and sodium could all pass through this one. It's normally closed unless this particular molecule, in this case acetylcholine, remember that's the neurotransmitter at neuromuscular junctions. That acetylcholine touches that and causes the channel to open, and now these ions can flow in or out through there. This is what happens at a neuromuscular junction. We didn't talk about it, but when a nerve impulse comes down, the neuron leading to a muscle fiber, the acetylcholine neurotransmitter is released, and it diffuses across that little gap. When those acetylcholine molecules touch ion channels in the surface of the muscle fiber, they open, and that allows positive, negative ions to flow in or out. That changes the electrical voltage on this plasma membrane, and that's what causes that impulse to spread out over the muscle fiber that causes it to contract. So that's what these ion-gated channels do. They open and allow ions, positive or negative ions, to flow in or out, and it changes the electrical voltage on the plasma membrane, and that's what leads to an action potential. The mechanically gated ones, again, some kind of mechanical pressure or stretching or something on this plasma membrane will cause that gate to open. Now ions can flow in or out. And the voltage-gated ones, change in voltage. But the word voltage just means the difference in electrical charge between two places, like on like outside the cell and inside the cell. And normally in a neuron, this is important, in a neuron, there's a lot of positive and negative ions outside and a lot of positive and negative ions inside. So there's sodium, potassium, calcium, chloride. But the combination of all those ions causes the electrical charge outside to be more positive and the electrical charge inside to be more negative. And that difference in electrical charge is this much, minus 70 millivolts. A millivolt is 1,000th of a volt, so these are very tiny, so it's not very much. But that's when So that's called the membrane voltage. Just the difference between the electrical charge outside and the electrical charge inside because of all the ions on both sides. And it's mostly because of sodium and potassium ions. A lot of positive potassium ions outside, and not as many positives. Or a lot of positive sodium ions outside, and positive potassium inside, but not as much as the positive sodium outside. And that's the voltage when there's no impulse, nerve impulse happening on this neuron. So if it's minus 70, normally, if these channels open and allow ions to flow through, if there's positive or negative ions moving in or out, that's going to change the membrane voltage or membrane potential. So now it's not minus 70 anymore. Now it's changed to minus 50 because of ions moving in or out. A summary of the same information there. This word polarized just refers to the difference in electrical charge inside compared to outside. So this is a neuron. It's hard to tell, but these are plus signs out on the outside and minus signs on the inside. So that's telling us that electrical charge is more on the outside, more positive outside and more negative inside, and that difference in electrical charge is minus 70 millivolts again, and there's no nerve impulse happening on this. So that's called the resting membrane potential. The word potential means the same thing as voltage. So the resting membrane potential of this neuron is minus 70 millivolts. That voltage changes if ion channels open and allow ions to flow in or out. It's just showing that if you had a little tiny voltmeter and you could measure that voltage difference, that voltage between the inside and outside. And this is the plasma membrane here. If you could measure the difference in electrical charge inside compared to outside, it would be minus 70 millivolts. And this is this is caused by, again, just mostly caused by a lot more positive sodium outside compared to positive potassium inside. But there's always, because of these leak channels, always some potassium leaking out and some sodium leaking in. And there's also these active transport channel proteins. Remember, active transport is a cell using energy to actually pump something in or out through the plasma membrane. And so these are called sodium-potassium pumps. These are just special channel proteins that use ATP for energy to pump something in or to pump something out or out of the cell or into the cell. In this case, these sodium-potassium pumps are always pumping. They'll pump 3 sodiums out for every 2 potassiums that they pump in. So these sodium potassium pumps are maintaining that difference, maintaining that resting membrane potential, having more sodium outside compared to potassium inside, so that the difference is minus 70 millivolts resting membrane potential. Okay, that's part one. Stand by for part two after break. It gets even more exciting. Part two. hardly possible. I can't promise that, but I'll try. I'll pull up a couple videos that we can watch that might be helpful.
My name is Trish. I'm 46 and I've struggled with depression most of my life. I have horrible seasonal depression.
This morning was a typical morning for me. I woke up thinking about that dream that I keep having about the guy in the sloth suit. And I got dressed because I was cold, and then I made some toast with butter because I was hungry.
What's that have to do with science? This we'll find out.
Not in one.
I don't know, they talking about some mess, talking about, Washington or Michigan had over 100% of votes, and how is that possible? Mm. I just think that 1 minute to go from seeing news.
What's on the bottom? Hold out.
There's one place, it says that the percentage was more than what it's supposed to be. They had a recount. It said there are more votes than registered voters.
Well, that's a mathematical problem, isn't it?
Right.
I mean, you can only have 100%.
Right, it's over 100%. It's one of those states, I forgot which one.
It's probably North Carolina. I mean, we just come out the box of one jackass.
No, it's either Michigan or Wisconsin. And Donald Trump is on my Instagram. He is got his lawyers turned up so that they can recount that state. Because that's the state that they want to flip. If they flip it, then Donald Trump wins. Not, then Biden wins. That's a flip state. You know, North Carolina and South Carolina are always a Republican state. So that state right there, he said that was a flip state.
I just wish America could do this properly. The whole world is watching us. Watching what absolute idiots we are together.
Right.
And they're just sitting, waiting
Right.
for an opportunity to strike. Some countries want nothing more than to have war on the world.
That's right.
They're just sitting, waiting.
Right.
Because they're like, these nitwits can't even get together and come back. How they gonna fight? How they gonna fight against us? They can't even figure out what they want to do.
Yeah.
Run this ESPN like 2 days ago. It says that literally nobody was working. Everybody was waiting to see when it was going to kick off polls, the early voting. That's all they are watching on TV. Us, America, what we're doing with this election. All they're watching.
Yeah, so they're going to see because
phones everywhere.
they're going to see. Mm-hmm. And the company that I work for is controlled by European people, Japanese people. Mm-hmm.
And they see they're just waiting, ground for us to give them a window of opportunity.
Mm-hmm. And this going to be the opportunity too.
And we're on display for the whole world to see, for the whole world to watch. You know? I wish we could just find a page. I don't know what page. Not a page, everybody get on.
It would never happen. It would never happen because this world is based off of different views and perspectives. Right. You know, and it will never change. They always say, let's make America great again. I never knew that it was great. You know, because that's just my opinion. I never knew that it was great because it seemed like when one president gets in the chair, he tries to do his best, and then it fails because people fail to realize that a lot of things that he put on the table, you have to get it signed and vetoed by the House. A lot of
And
things.
then from the House goes to the Senate, and the Senate says no, it's not.
Right, that's what I'm saying, but a lot of people don't know that. They always blame the president.
And they are just, puppets.
Right.
They're nothing more. They don't have to follow something.
Right. You know,
I remember traveling abroad and we were in Amsterdam. That's my best time ever. And then where we were, it's down that and their whole outlook, their whole
I'm recording this, so make sure.
I mean, they still were very nice to us. We still had like wonderful accommodations. You know, like, one of them, I think I don't know when the last week they said, it was like, you know, why do y'all live here?
that's how my father, he was in peace.
Could not figure out why in the world. And I'm like,
Yeah.
I live somewhere.
Yeah.
The whole honest, I know I don't.
Our country is free. It's not like a lot of countries.
It's so serious.
Right. It's like they're really divided. Well, you know, right. And I learned the Constitution, the preamble. I learned that in high school, and no, yeah,
Maybe one day we really will, baby.
right.
It don't look like it's gonna be years.
No.
Let's hope for a better year next.
Right.
Yeah, so it's either we get them out, but they don't have to.
I told you they got the 3 pounds.
They got over 100%.
Told you. I just told you they didn't.
Okay, so that would
No, what it was is they said on the news that the votes was more than what the voters registration. And the lady from CNN said that they didn't know that they had that many people live here in Wisconsin. They had more people.
I had a census taker knock on my door once.
Yeah, they have
Every
more.
week for the last 2 months. I
Yeah.
didn't go up there and work.
Yeah, they said 2 states that was like that. Mm-hmm.
I thought one lady was gonna fall and break a hip in my yard. She was trying to come up on.
But you see it, though,
I was
don't
like,
you?
lady, get off my property. Ew.
See it though, don't you?
Mm-hmm. They don't have, they didn't do a proper census. Oh wait, this says a lot of people did a mail-in ballot, but the mail-in ballots never arrived and got tallied, so people showed up to do same-day voting. And when they went to vote, it didn't show up that they had done a mail-in ballot. Where are all those mail-in ballots that were done?
But Biden the one that wants to go to the state. You know what I'm saying? It's blue. Both of those states are blue. Either I'm gonna go with the flow. That's fine.
Till it's more North Carolina. Sixteen states have dispatched the National Guard to allow people to vote. For years, the National Guard has to stay out there and allow people to go vote. 1960 all over again. 16 states.
I wonder how much they got in the,
It's, I look through,
uh,
it's on the 14th, this one, when they do the phones and stuff. Right now, they doing the 65 and the 55. 55 and 228 and the 65.
how much?
228. I know Rockingham got them now. It's still
Yeah, 55. Yeah, 55, 240,
228.
228. Same thing, brand, all brand. On there, got, 228. The 55 in Campbell and Rockingham. Okay, well, we gonna put it.
And they give you stuff in themselves that they talk
Well, we
about.
want
Y'all remember Black Friday used to be one day? It's because every, Walmart when we close Black Friday. It's Thanksgiving.
Okay, when I get home.
I was like, so what people gonna do at Thanksgiving Day?
Alright.
Maybe some eggs, cheese, but.
I wanna know how much,
You're gonna put it on whatever. Whatever meal for Thanksgiving? No. I'm gonna do retail, ain't nobody gonna be
yeah, for that.
they got a little bitty grocery store in there.
I just can't wait to do some dressing. I feel like this all the time of year, like we did. Some dressing, some jiffy brand here.
Okay.
What day what day first?
Saturday, November 14th, it's gonna be online at 12, and it's still at 5. And then do it twice.
Jamie, Tatum. Hard taking attendance when people are wearing masks sometimes. Somebody's student came to my office this afternoon and said he was looking for Mr. Beck. And I was sitting in there by myself, so I had my mask off. And when I put my mask on, then he recognized me because he had never seen me. He had never seen me without the mask on. Okay, let's do part 2. I think it's a little shorter. I want to show a couple of videos at the end that I hope might clarify or confuse you further. Okay, we already saw this or something like it. Okay, before we describe an action potential, which is an action potential is the nerve impulse that conducts along the axon of a neuron. But a graded potential is can happen anywhere on a neuron, and it's just a graded potential just means a change in the membrane voltage will cause beta channels open and allow positive or negative ions to flow in or out. So it just changes the membrane voltage so that it's not minus 70 millivolts anymore. It could be minus 80 or minus 90, or it could go the other way and be minus 60, minus 50, or even become positive because of ions flowing through. And we'll see that graded potentials cause action potentials to happen. They're called graded potentials because just like your grade in this class can be high or low, a graded potential can be large or small. It could be a large change in the membrane voltage, or it could be a large change or a small change. Change it a lot or just a little bit. But we'll see that action potentials they're all the same size. There's no big ones or small ones. Action potentials either happen or they don't, and when they happen, they're all the same size. This is showing what happens to the membrane voltage in an action potential. So this always begins at the axon hillock. So let's look at a picture. Action potential always begins at the axon hillock. So right where the axon connects to the cell body, that's where an action potential begins, and then it conducts along the axon to the other end. And what happens there is this change in the membrane voltage, which when there's no nerve impulse, and we call it the resting membrane potential, or membrane voltage, is minus 70 millivolts, more positive outside of the neuron and more positive inside. But then something happens that changes the voltage, so it becomes less negative and less negative, and if it reaches this level of minus 55, that's called the threshold. The threshold voltage. The threshold is like this, like the bottom of a doorway. You step over it. Once you get over it, now you're into the room. But so once the voltage changes and it reaches this level, minus 55 millivolts, that will cause an action potential to happen along that axon. And why is that there are voltage-gated ion channels in the plasma membrane at the axon hillock. And so when the voltage at those ion channels reaches minus 55, they open. And that allows a lot of positive sodium ions to flow in, change the voltage even more. We got more positive stuff coming in. So instead of being more positive outside, negative inside, now it becomes more positive inside, negative outside. Because all those positive sodium ions flowing in. And so that's what's causing this membrane voltage or potential to become less and less negative, goes all the way to zero, and even goes up to about positive plus 30 millivolts. That's called depolarization. So instead of the membrane being polarized, so it's positive outside, negative inside, it depolarizes all the way to zero and even goes positive. And those ion channels for sodium stay open for just a brief moment and then they close. And then potassium channels open, and now positive potassium ions flow out to make it positive outside, negative inside again. And that's what causes this repolarization. It even goes a little bit beyond minus 70 and then, but then it evens out to minus 78. So, so then it's back to resting membrane potential. And that happens, that starts at the axon hillock. That depolarization and then repolarization starts at one end of the axon, at the axon hillock, and then it just moves along to the other end. Because those changes in voltage just cause more voltage-gated ion channels a little farther down to open. That causes the ones a little farther down to open. And so there's just this wave of depolarization and then repolarization happens all along that axon. And that all happens in just a tiny fraction of a second. It's just lightning fast. So what causes this? What causes this change in the membrane potential from to go from minus 70 to minus 55 in the first place? It's graded potentials on that neuron. So here's a picture of a. So if there are graded potentials anywhere on that neuron that cause ion channels to open and positive or negative ions to flow in or out, that's going to cause a change in the membrane potential on that neuron. When that change in membrane potential reaches the axon hillock, if that causes that membrane potential at the axon hillock to become minus 55, then those sodium ion channels open and it causes an action potential. If those action potentials can be large or small, and they can cause the membrane potential to go either way, either direction. It could, like this one, this graded potential starts out at minus 70, but it goes more negative. It goes down to like minus 73 or something. So that's called a hyperpolarizing graded potential. It just makes it more polarized. Membrane more polarized. But it could also be a graded potentials can also be depolarizing, like this one. So it goes from minus 70 up to about minus 60. So that's getting closer to minus 55. It goes all the way to minus 55. That's the trigger for an action potential. And whether it's hyperpolarizing or depolarizing, it just depends on whether it's positive ions flowing in or negative ions flowing out, or so whether it makes the membrane either more positive outside compared to negative inside, or less positive outside, less negative inside. So what causes these graded potentials? It depends on what kind of neuron it is. It can, like we've seen, it can be neurotransmitters at a synapse where another neuron contacts a neuron, could cause ion channels to open. It could be if it's some kind of touch receptor or pressure receptor neuron, then some kind of touch or pressure could cause ion channels to open, and ions flow in or out, cause a nerve impulse to go to the brain, telling your brain that there's some pressure at that part of the body. And so these graded potentials can be caused by internal changes. If it's some kind of sensory receptor, that's measuring, for example, measuring the amount of oxygen in your blood. The oxygen in your blood could cause ion channels to open or close on neurons. It would cause an action potential to go to your brain. Or blood pressure. The pressure of your blood pressure could cause these blood pressure sensory receptors in some blood vessels to stretch, and that stretching would cause ion channels to open. Ions flow in or out and cause a graded potential that would cause an action potential to go to the brain, sending information to your brain about your blood pressure. Or it could be external stimuli. So, like I've mentioned, touch, some kind of pressure on your skin, and there's neurons there that are sensitive to pressure that causes ion channels to open. Could be vision. In the retina of your eyes, you have they're called photoreceptors. When light hits them, it causes ion channels to open. Ions flow into the neurons, cause nerve impulses to go to the brain. Smell, taste, sound. These smell and taste those would be ligand gated. So certain kinds of chemicals in the air you're inhaling, or in the chemicals in the food you're eating, when they come into contact with these ion channels on the receptors, would cause ion channels to open. Ions flow in or out, cause a graded potential, which would cause an action potential to go to your brain. And again, same thing we've seen before. So these are just gated ion channels that open. In some cases, because of some kind of mechanical or physical pressure or stretching or vibration. In other cases, of some kind of chemical that comes into contact with them, causes them to open, and ions flow in or out, causing a graded potential. So, for example, this is normally when these gates are closed, positive outside, negative inside, difference of 70 millivolts. If these open, and now there's some positive ions flowing in. Now some of this positive stuff is going in, so it'll be less positive outside, more positive or less negative inside. It's going to change the voltage. That's what a graded potential is. Just a change in the voltage will cause ions move in or out. This is showing that graded potentials can be big or small. So to change the voltage a little bit from like minus 70 to minus 66 or something, and bigger ones would be change it a little more to minus 62 or something. That one's like all the way to minus 60. And it just depends on the stimulus, how much of a stimulus. So maybe, for example, if I just let me touch my fingers, it would cause some ion channels to open, change the voltage just that much. But if a little more pressure would cause more ion channels to open, maybe it would stay open for a longer time. More ions would flow in or out, cause more of a change, or even harder pressure, bigger stimulus, bigger change in the membrane voltage. If that change is enough to reach all the way to minus 55 at the axon hillock, then an action potential would happen along that axon of that neuron. And this shows that graded potentials can add together. So, well, if there's there's more than one stimulus that happen together, at least either one right after another, real close together in time, or real close together physically. And those two graded potentials, if they're on the same neuron, they could add together. And if their added effect causes the membrane potential to reach minus 55, then it would cause an action potential. This is showing part of an axon that has an action potential moving along it. So here's normal resting potential, positive outside, negative inside, except here it's negative outside, positive inside, because this action potential, it started here and it's moving along the axon, and right at this moment it's here. So this is a reverse in the polarization. It's depolarized right here. This action potential has already moved past here, and it's been repolarized. It's back to resting potential. So this just starts at the axon hillock. It just moves rapidly along the whole axon. This rapid depolarization of the membrane and then repolarization happens because if the voltage, membrane voltage here at the axon hillock, the beginning of the axon, if it reaches minus 55 because of graded potentials, that will cause positive sodium ions to open. Positive sodium flows in, changing it to positive inside, negative outside. And then a moment later, those sodium channels close and positive potassium ion channels open. It allows positive potassium to flow from the inside out to repolarize the membrane. And meanwhile, those sodium-potassium pumps are pumping sodium in and potassium out. Sodium out and potassium in. That'll help return the membrane voltage to resting minus 70. So this all happened just in a fraction of a second along the axon. Okay, so this describes all this in steps. So resting starts out at resting potential minus 70, and then one or more graded potentials happens on that neuron that change its membrane potential. And if that causes the membrane potential at the trigger zone, at the axon hillock, to reach minus 55, then that's the trigger for an action potential to conduct along that axon. The membrane potential will be depolarized all the way to plus 30, and then immediately go right back to minus 70. But that change will start at one end, at the axon hillock, and it'll just conduct along the axon to the other end, and then it's over. Back to resting membrane potential. Okay, shows it again here. So at the top, that's at resting potential. Positive output, it's more positive outside, negative inside, because there's way more sodium, positive sodium outside than there is positive potassium inside. And there's other ions involved also. There's calcium ions and fluoride, negative fluoride ions, but it's mostly sodium and potassium. So when if it reaches minus 55 millivolts here, because of graded potentials on that neuron, then these sodium ion channels open, so a lot of positive sodium flows in. that makes it, that depolarizes it. So instead of being positive outside, negative inside, now it's negative inside, negative outside, positive inside. And a moment later, those sodium channels close and the potassium channels open. And now there's positive sodium or positive potassium flowing out, putting it back to positive outside, negative inside. So it's very rapid depolarization and then repolarization. And because of the change in voltage here, as these channels are opening and closing, it causes the same thing that happened here. It causes these sodium channels to open and then close, which causes these to open and close. So these ion channels opening and closing causes more change in the membrane potential, which causes these ion channels to open, causes change in membrane potential here, which causes these to open. And so it just conducts itself all along the axon. And then it goes back to resting membrane potential of minus 70. It's always, resting potential is always minus 70, and and an action potential always goes up to about plus 30. So there's no big action potentials or small action potentials. If it happens, it'll be all the way. So graded potentials can be big or small, but the action potentials, they're all the same size. If this reaches minus 55, this is going to go up to about 30 and back down. What determines if, for example, if there's a loud sound compared to a soft sound coming into your ears, how does your brain know if it's loud or soft? It's not because a loud sound will cause your ears to send a bigger action potential to your brain. It'll send more action potential per second. It's the frequency of the action potentials that tells your brain how the size of that stimulus. So if it's a very faint sound, your ears might send just 10 action potentials per second to your brain. But if it's a louder sound, it might send 100 action potentials to your brain. But each action potential, they're all the same size. It's just how many per second can vary. Okay, so a stimulus that is not enough to reach minus 55 millivolts is called a subthreshold. It just means below the threshold. It doesn't bring the voltage to minus 55, so there would not be an action potential. So here's it's kind of showing that about the stimulus is not enough to cause the membrane potential to reach minus 55, then there will not be an action potential. If it is enough, if it reaches minus 55, then it'll shoot up to plus 30. There will be an action potential. If there's a really strong stimulus, then it won't cause bigger action potential. It'll just cause several in a row real fast. Action potentials. Okay, this is the same information again, just another set of steps and diagrams showing the same thing. So this is at resting potential, minus 70 millivolts. And the sodium and potassium ion channel, they're both closed. But then if the graded potentials cause it to reach minus, the membrane voltage here to reach minus 55, then the gate opens. Now a lot of positive sodium's flowing in, and now it's positive inside, negative outside, instead of negative inside, positive outside. So the membrane voltage depolarizes. It becomes less and less negative, all the way to 0, and it goes all the way up to 30. And then a moment later, the sodium channels close. Now the potassium channels open, so positive potassium ions flowing out, and that'll repolarize the membrane. Just so exciting you can't contain yourself, or okay, then it goes back to resting potential. So this is, they're all or none. That means there are no big action potentials or small action potentials. It either happens or it doesn't, and they're all the same size. This continuous versus saltatory conduction, that's comparing the an axon that has myelin sheaths to ones that don't. So if there's no myelin on the axon, that's called continuous conduction. The action potential just goes all along here. But if there's myelin sheaths, then the action potential, it goes along these bare sections, but then it skips over, it jumps over to the next gap. And so that makes it faster. Action potentials are faster if there's myelin than if there's no myelin. It's called saltatory conduction. Salt is Latin for jumping, so it jumps over the myelin sheaths. Did I do that? The things that speed up an action potential. Of course, myelination, we just said, but also the axon diameter. So thicker axons have faster action potentials. If this axon was thicker than this one, then this would have faster action potentials. But the myelin is actually more important than the thickness. So even if this axon with the myelin was the same thickness as this axon without myelin, this would still be faster. So the myelin is more important than the thickness of the axon. And also temperature. At higher temperatures, action potentials are faster. And conversely, at lower temperatures, they're slower. So this is one reason that ice on an injury will reduce the pain sensation, because it slows down the action potentials that go to your brain. It's also why if you're outside in the cold for a long time and you become hypothermic, so your body temperature is actually dropping lower than normal, your body functions slow down. You lose coordination. People start to stumble and fall. They can't use their fingers very well. Okay, synapses. A synapse, where the kind that we've been talking about, where there's neurotransmitters released, like this one, this kind of synapse. So here's one neuron, the axon terminal, one neuron, and then this would be this could be the cell body or the dendrite of another axon here, or this could be a muscle fiber here. So this could be a neuromuscular junction. But if it's this kind of synapse where there's neurotransmitters released at the synapse, and then the neurotransmitters move across the gap and they come into contact with receptors. These receptors are just ligand-gated ion channels. So this is what happens at a neuromuscular junction. These ligand chemical-gated ion channels open and ions flow through here to change the membrane potential on the muscle fiber, and that's what causes that impulse to spread out over the whole muscle that causes it to contract. But this is called a chemical synapse because it involves neurotransmitters. But there's another kind of synapse called electrical synapse. And so with these electrical synapses, there's just two neurons side by side, and they're just tightly connected. There's no neurotransmitters. There's no gap in between. They're just touching. So an action potential comes down one and just continues right along the next one. There's no with this. It takes a little bit of time for these, and the nerve impulse action potential reaches here. It causes these vesicles to move to the surface and really release the neurotransmitters. Neurotransmitters drift across here, contact these ion channels. The ion channels open and ions flow in, and it causes a graded potential here that could cause an action potential in this next neuron. It only takes a fraction of a second for that to happen, but it does take time. These electrical synapses, it takes no time at all. Just action potential just comes along one neuron, just continues on to the next one. These electrical synapses are what are at the intercalated discs in cardiac muscle fibers. Remember those, for the cardiac muscle fibers are connected end to end. It holds them together, but it also allows the impulse to come from one cardiac muscle fiber, and then it just continues on to the next one. And that way, that impulse that starts at the pacemaker in your heart, it spreads out to all your cardiac muscle fibers in that heart chamber so that they all contract almost at the same moment. So here's just showing that these synapses, which are connections between neurons, it can be from like this case, an axon from one neuron has a synapse with the dendrites of this other neuron. Here, this axon from one neuron forms a synapse right on the cell body of this other neuron. And in this case, this axon is forming a synapse right at the axon hillock. So there's all these different kinds of arrangements or locations of where these synapses can be between different neurons. When there's a synapse between neurons, the information goes in this direction, so along this axon, into this neuron, and then out this axon. So this neuron would be called the presynaptic neuron. Pre means before, so it's before the synapse. This one would be called the postsynaptic, or after the synapse neuron. The same with this. That's the presynaptic neuron. This would be the postsynaptic neuron. That's the presynaptic neuron, and this would be the postsynaptic one. Again, this would be the presynaptic one. There's a synapse here, so here's the presynaptic one, and this would be the postsynaptic neuron. So that leads us to these terms, these big long terms: excitatory postsynaptic potential, EPSP, and inhibitory postsynaptic potentials. Those are big long terms, but they're really they're really pretty simple. Postsynaptic means it happens to the postsynaptic neuron, the one after the synapse. And potential just means graded potential. So so this would be the presynaptic neuron and the postsynaptic neuron. And here's the synapse. And so when the nerve impulse comes along this neuron, neurotransmitters are released. They cause the neurotransmitter molecules come into contact with these ligand-gated ion channels, which open up and allow, in this case, positive sodium to flow in. That's going to change the membrane potential on this postsynaptic neuron. That's the graded potential, just a change in the membrane potential on a neuron. So if that graded potential would be depolarizing graded potential because of sodium ions flowing in, then we would call that an excitatory postsynaptic potential. Excitatory because it's depolarizing, which means it's going to change that membrane potential from -70 to something less than that, -65, -60, -55. So it's getting closer to the threshold potential of -55. That's why it's excitatory. It means it's making it more likely that there's going to be an action potential. It's getting closer to -55. If it goes the other way, hyperpolarizes that postsynaptic neuron, so it makes its membrane potential -75, -80, -90, getting farther away from the threshold of -55. It's inhibitory because it's inhibiting action potential, making it less likely that there's going to be an action potential in that postsynaptic neuron. Because its membrane potential is getting farther away from -55. So these action potentials, some of them are inhibitory, some are excitatory, and if there are inhibitory and excitatory ones happening at the same time to a neuron, they combine. Their combined effect, whatever their combined effect is at the axon hillock, if it reaches -55, there will be an action potential. If there's not, there won't be. So this spatial and temporal summation just means combining the graded potential. Spatial summation means, spatial means in their location in space. So if there's, if there's, if this neuron here is receiving a lot of different graded potentials on those different dendrites all at the same time. Some are inhibitory, some are excitatory. They just all add together. And whatever their effect is at the axon hillock, if it reaches -55, there will be an action potential. That would be this spatial summation. Where am I? Spatial summation. Just adding them up, or combining the effects of graded potentials at different places. But the temporal or time summation means if there are a lot of graded potentials on the same neuron close together in time, like several within 1 second, but either in the same place on the neuron or in different places, they'll still can be added together, and their effect cause action potential or not. neurotransmitter molecules at a synapse, once they're released, so once these neurotransmitter molecules are released, here, if there's more and more action potentials causing more and more neurotransmitters to be released, they won't just accumulate in here. They something happens to remove them, and that can be it can either just diffuse out, drift away, or there can be enzymes. In some cases, there are enzymes that will break them down, and in some cases, they'll be taken back up. They'll be taken back up into the neuron, or there will be neuroglia, which will take the neurotransmitter molecules and just like absorb them, and then recycle them. You may have heard of SSRIs. Anybody know what SSRI drugs are? They're antidepressants. It stands for selective serotonin reuptake inhibitor. Serotonin is one of the neurotransmitters at some synapses in the brain, and these drugs, they inhibit the reuptake of serotonin at these synapses, so it allows serotonin to accumulate more in these synapses. And serotonin is a neurotransmitter that just makes you feel better. So this just causes more stimulation or more nerve impulses involving serotonin in the brain, which has the effect of reducing depression. Let's skip over some of this stuff. Okay, there's many different chemicals in the body that function as neurotransmitters in the many different synapses. We learned about that first one, acetylcholine. That's always the neurotransmitter at neuromuscular junctions. It's always acetylcholine. But at other synapses, there's many different kinds of synapses in the billions of neurons in our nervous systems that use these different. Some of these you've certainly heard of: dopamine, serotonin, endorphins. Some of these also function like norepinephrine. That also functions as a hormone, but it's so that's one of the fight or flight hormones. Epinephrine and norepinephrine, also called adrenaline and noradrenaline, but it also functions as a neurotransmitter at some sense. Okay, well, just more about neurotransmitters. You don't need to know what each one does or anything. This concept called plasticity or neural plasticity. The word plasticity just means the ability to change. Something is plastic, it means it can change form. Neuroplasticity just means that your nervous system can change. Your central nervous system, especially your brain, can change. How your brain functions has more to do with the connections among your neurons than how many neurons you have. And so you can change your brain by exercising it in different ways and causing new connections between the neurons that you have. And just by being a student in a difficult class like this, you are changing your brain. Your brain is learning how to learn. I hope you'll find that as you go through school. It certainly did to me. I feel like I got way smarter. Not only accumulated knowledge, but my brain just got smarter as I went through college. I was just able to learn better. This plasticity is used in treatment or therapy of people who have brain damage, either from a stroke or from a traumatic brain injury. So a large part of their brain is just gone. neurons are dead, destroyed. They won't grow back. But you can grow new connections between the neurons that you have left. And so stroke patients and TBI patients, traumatic brain injury patients, are if they are treated by a therapist who knows how to exercise their brains to make the new connections to restore some brain functions that were lost, and they can recover a lot of those functions that had been done by the neurons that are gone. The remaining neurons can learn how to, by making new connections, can restore some of those functions. Regeneration, ability to replicate or repair. The peripheral nervous system can do this way better than the central nervous system. You have a nerve damage in your central nervous system, your brain or spinal cord, you won't regrow those neurons, or you won't be replaced by cells dividing to make new ones. Like I mentioned earlier when we talked about the Schwann cells make the myelin on the cells of the peripheral nervous system, but they also function in regenerating damaged axons or severed or cut axons. They'll help to regrow them. So that's what's shown here. The Schwann cells make what's called a regeneration tube, and then which is made of myelin, and then it'll grow a new axon inside there. Okay. Big long chapter. Any questions about that stuff? You need to just go over this stuff a few hundred times for it to sink in and understand how it all fits together. It all makes sense, but there's a lot to absorb, I know. So let's look at these videos and then get out of here. Where is it? Oops, sorry.
Keep having about the guy in the slop. This morning was a typical morning for me. I woke up thinking about that dream that I keep having about the guy in the sloth suit. And I got dressed because I was cold, and then I made some toast with butter because I was hungry, and then I let the dog out because she was whining and staring at me. And then I made some tea, but I let it cool off before I drank it because it burned my mouth yesterday. In addition to being just part of my morning ritual, all of these actions are examples of what my nervous system does for me. A weirdo dream, the sensation of cold air and hot tea, deciding what to put on the toast, going to the door, the sound of the dog, all that was processed and executed by electrical and chemical signals to from nerve cells. You can't oversell the importance of the nervous system. It controls all the things, all your organs, all your physiological and psychological reactions, even your body's other major controlling force, the endocrine system, bows down before the nervous system. There is no you without it. There's no me without it. There's no dogs without it. There's no animals. There's no things. There's things. It's important. That's why we're dedicating the next several episodes to the fundamentals of the nervous system, its anatomy and organization, how it communicates, and what happens when it gets damaged. This is Mission Control, people. Even though pretty much all animals, except super simple ones like sponges, have a nervous system, ours is probably the most distinctive feature of our species. From writing novels to debating time travel to juggling knives, all of your thoughts and actions and emotions can be boiled down to three principal functions: sensory input, integration, and motor output. Imagine a spider walking onto your bare knee. The sensory receptors on your skin detect those 8 little legs. That information is your sensory input. From there, your nervous system processes that input and decides what should be done about it. That's called integration. And should I be all zen about it and just let it walk over me? Or should I not be zen and freak out, run around screaming, Spider! Your hand lashing out to remove the spider and maybe your accompanying banshee scream is the motor output, the response that occurs when your nervous system activates certain parts of your body. As you can imagine, it takes a highly integrated system to detect, process, and act on data like this all the time. And when we talk about the nervous system, we're really talking about several levels of organization, starting with two main parts, the central and peripheral nervous systems. The central nervous system is your brain and spinal cord, the main control center. It's what decided to remove the spider and gave the order to your hand. Your peripheral nervous system is composed of all the nerves that branch off from the brain and spine that allow your central nervous system to communicate with the rest of your body. And since its job is communication, your peripheral system is set up to work both directions. The sensory or afferent division is what picks up sensory stimuli, like, hey, there's an arachnid on you, and slings that information to the brain. Your motor or efferent division is the part that sends directions from your brain to the muscles and glands, like, hey, hand part, how about you do something about that spider? Motor division also includes the somatic or voluntary nervous system that rules your skeletal muscle movement, and the autonomic or involuntary nervous system that keeps your heart beating and your lungs breathing and your stomach churning. And finally, that autonomic system too has its own complementary forces. Its sympathetic division mobilizes the body into action and gets it all fired up like, ah, spider, while the parasympathetic division relaxes the body. It down, like it wasn't a black widow or anything. Fine, breathe. So that's the organization of your nervous system in a nutshell. But no matter what part you're talking about, they're all made up of mainly nervous tissue, which you'll remember is densely packed with cells. Maybe less than 20% of that tissue consists of extracellular space. Everything else, cells. The type of cells you most likely heard of are the neurons, or nerve cells, which respond to stimuli and transmit signals. These cells get all the publicity. They're the ones we're always thanking every time we ace an exam or think up a snappy comeback to an argument. But these ones, guys, really account for just a small part of your nervous tissue because they are surrounded and protected by gaggles of neuroglia, or glial cells. Once considered just the scaffolding or glue that held neurons together, we now know that our different glial cell types serve many other important functions, and they make up about half of the mass of your brain, outnumbering their neuron colleagues by about 10 to 1. Star-shaped astrocytes are found in your central nervous system and are your most abundant and versatile glial cells. They anchor neurons to their blood supply and govern the exchange of materials between neurons and capillaries. Also in your central nervous system are your protective microglial cells. They're smaller and kind of thorny looking and act as the main source of immune defense against invading microorganisms in the brain and spinal cord. Your ependable cells line cavities in your brain and spinal cord and create, secrete, and circulate cerebrospinal fluid that fills those cavities and cushions those organs. And finally, your central nervous system's oligodendrocytes wrap around neurons, producing an insulating barrier called the myelin sheath. Now, over in your peripheral nervous system, there are just two kinds of glial cells. Satellite cells do mainly in the peripheral system what astrocyte cells do in the central system. They surround and support neuron cell bodies, while Schwann cells are similar to your oligodendrocytes in that they wrap around axons and make that insulating myelin sheath. So don't sell your glial cells short. They're in the majority cell-wise, but of course when it comes to passing tests and winning arguments, most of the heavy lifting is done by the neurons. And they're not all the same. They're actually highly specialized, coming in all shapes and sizes, from tiny ones in your brain to the ones that run the entire length of your leg. But they do all share three super cool things in common. Number one, they're some of the longest-lived cells in your body. There's a lot of debate right now about whether you're actually born with all the neurons you'll ever have, but some research suggests that at least in your brain's cerebral cortex, your neurons will live as long as you do. Cool fact number two, they are irreplaceable. It's a good thing that they have such longevity because your neurons aren't like your constantly renewing skin cells. Most neurons are amitotic, so once they take on their given roles in the nervous system, they lose their ability to divide. So be careful. And number three, they have huge appetites. Like a soccer-playing teenager, neurons have a crazy high metabolic rate. They need a steady and abundant supply of glucose and oxygen, and about 25% of the calories that you take in every day are consumed by your brain's activity. Along with all these wonderful qualities, your neurons also share the same basic structure. The soma, or cell body, is the neuron's life support. It's got all the normal cell goodies, like a nucleus, DNA, mitochondria, ribosomes, cytoplasm. The bushy branch-like things projecting out from the soma are dendrites. They're the listeners. They pick up messages, news, gossip from other cells, and convey that information to the cell body. The neuron's axon, meanwhile, is like the talker. This long extension or fiber can be super short or run a full meter from your spine down to your ankle. We've got a few different axon layouts in our body, but in the most abundant type of neuron, the axons transmit electrical impulses away from the cell body to other cells. For us students of biology, it's a good thing that nerve cells aren't all identical because their differences in structure are one of the ways that we tell them apart and classify them. The main feature we look at is how many processes extend out cell body. A process, in this case, being a projecting part of an organic structure. 99% of all your neurons are multipolar neurons with 3 or more processes sticking out from the soma, including one axon and a bunch of dendrites. Bipolar neurons have 2 processes, an axon and a single dendrite, extending from opposite sides of the cell body. They're pretty rare, found only in a few special sensory places like the retina of your eye. Unipolar neurons, on the other hand, have just 1 process and are found mostly in your sensory receptors. So if you ever find yourself probing around someone's nervous tissue, remember these 3 terms to help you figure out what you're looking at. So because we're talking about physiology here as well as anatomy, we have to classify these cells in terms of their function. And that basically comes down to which way an impulse travels through a neuron in relation to the brain and spine. Our sensory, or afferent neurons, pick up messages and transmit impulses from sensory receptors in, say, the skin or internal organs, and send them toward the central nervous system. Most sensory neurons are unipolar. Motor, or afferent neurons, do the opposite. They're mostly multipolar and transmit impulses away from the central nervous system and out to your body's muscles and glands. And then there are interneurons, or association neurons, which live in the central nervous system and transmit impulses between those sensory and motor neurons. Interneurons are the most abundant of your body's neurons and are mostly multipolar. Okay, it's applied knowledge time. Let's review everything we've learned so far in terms of that spider on your knee. Those 8 creeping legs first activate your unipolar sensory neurons in the skin on your knee, and they send something crawling on you. The signal travels up an axon wrapped in Schwann cells and into your spinal cord, where it gets passed on to several multipolar interneurons. Now, some of those interneurons might send a signal straight down a bunch of multipolar neurons to your quadriceps muscle on your thigh, triggering you to kick your leg out before you even know what's going on. Other interneurons will pass that signal to neurons that carry it up your spinal cord to your brain. That's where your body first recognizes that thing as a spider, and the connections between neurons interpret and split the signal so that you can either scream and start swinging your arms wildly about, or remain calm and with dignity remove the spider from your person. It's all based on the connections between neurons, which brings me to a whole new question. How? How in the name of Jean-Martin Charcot do nerve cells use chemistry and electricity to communicate with each other? It's one of the most stupefyingly awesome and complicated aspects of your nervous system and basically of all life, and it is what we will cover in our next lesson. Today, we learn how sensory input integration and motor output of your nervous system basically rules your world. We talked about how the central and peripheral...
At American Public University, we believe together we can achieve more. So we provide
What if everything you did, thought, and felt could be communicated by pushing a button? It'd be like the world's simplest app, one that just sends out a little ping, always at the same volume and length, to communicate everything from "it sure is cold in here" to "I love churros" to "boy, I sure would like to breathe sometime soon." Well, that is actually exactly how your neurons send all the impulses responsible for every one of your actions, thoughts, and emotions. When a neuron is stimulated enough, it fires an electrical impulse that zips down its axon to its neighboring neurons. But they've only got one signal that they can send, and it only transmits at one uniform strength and speed. What they can vary is the frequency or number of pulses. Like this is distinct from this. And your brain can translate these signals, reading them like binary code, organizing them by location, sensation, magnitude, and importance, so that you know the difference between turn up the thermostat and oh my gosh, I'm on fire. That buzz, that nerve impulse, is called the action potential. It's one of the most fundamental aspects of anatomy and physiology and really life in general. It's happening inside of you right now, and we want to make sure that you understand what all that buzz is about. Before we delve into how neurons communicate, we've got to first understand a little bit of our old friend electricity. Basically, your body is a sack of batteries. No, I mean, you don't look like a sack of batteries. I'm just saying that your body as a whole is electrically neutral with equal amounts of positive and negative charges floating around, but certain areas are more positively charged than others. Because opposite charges attract, we need barriers or membranes to keep positive and negative charges separate until we're ready to use the energy that their attraction creates. In other words, we keep them separated to build potential. A battery, just sitting on its own, has both a positive and a negative end, and the potential to release energy. But it doesn't do anything until it's hooked up to a flashlight or a phone or a kid's toy that lets those charges move toward each other, on the way converting electricity into light or sound or children's laughter. In much the same way, each neuron in your body is like its own little battery with its own separated charges. It just needs an event to trigger the action that brings those charges together. If you're thinking that this sounds more like engineering than anatomy, that might not be a bad thing. It might even help to think of your neurons in the same terms an electrician might use. Voltage, for example, is the measure of potential energy generated by separated charges. It's measured in volts, but in the case of your body, it's millivolts. Small amount. In a cell, we refer to this difference in charge as the membrane potential. The bigger the difference between the positive and negative areas, the higher the voltage and the larger the potential. And just like there's voltage in your body, there's also current, the flow of electricity from one point to another. The amount of charge in a current is related both to its voltage and its resistance. Resistance is just whatever is getting in the way of the current. Something with a high resistance is an insulator, like plastic, and something with a low resistance, a conductor. Now, when we talk about these concepts in terms of you, we're typically talking about how currents indicate the flow of positively or negatively charged ions across the resistance of your cell's membrane. And again, these membranes separate the charges, so they're what provide the potential to convert the electricity into something useful. Okay, now that we got electricity 101 down, let's see how it works inside your nervous system. A resting neuron is like a battery sitting in that sack that is you. When it's just sitting there, it's more negative on the inside of the cell relative to the extracellular space around it. The difference is known as the neuron's resting membrane potential. It sits at around negative 70 millivolts. Where do those charges come from? Outside of a resting neuron, there's a bunch of positive sodium ions floating around, just lingering outside the membrane. Inside, the neuron holds potassium ions that are positive as well, but they're mingled with bigger negatively charged proteins. And since there are more sodium ions outside than there are potassium ions inside, the cell's interior has an overall negative charge. When a neuron has a negative membrane potential like this, it is said to be polarized. Now, these ions didn't just show up in this arrangement on their own. This is all orchestrated by one of the most important bits of machinery in your nervous system, the sodium-potassium pump. This little protein straddles the membrane of the neuron, and there are tons of them all along the axon. For every 2 potassium ions it pumps into the cell, it pumps out 3 sodium ions. This creates a difference in the concentration of sodium and potassium, and a difference in charges, making it more positive outside the neuron. This difference is an electrochemical gradient. You probably know enough about biology by now to know that nature hates gradients. It wants to even out all of those inequalities in concentration and in charge to restore balance. But the only way to even out that gradient is for the ions to pass across the membrane. Thankfully, the sodium-potassium pump isn't the only way in or out of the cell. The membrane is also riddled with ion channels, large proteins that can provide safe passage across the membrane when their respective gates are open. And these gates open and close for different reasons depending on their structure and purpose. Most are voltage-gated channels, which open at certain membrane potentials and close at others. For example, sodium channels in your neurons like to open around negative 55 millivolts. Some others are ligand-gated channels. They only open up when a specific neurotransmitter, like serotonin or a hormone, latches onto it. Then we also have mechanically gated channels, which open in response to physically stretching the membrane. In any case, when the gates do open, ions quickly diffuse across that membrane down their electrochemical gradient, evening out the concentrations and running away from other positively charged ions. This movement of ions is the key to all electrical events in neurons, and thus is the force behind every single thing we do and feel. Of course, not all of your body's electrical responses are the same, and neither are the flows of ions going in and out of your neurons. If only a few channels open and only a bit of sodium enters the cell, that causes just a little change in the membrane potential in a localized part of the cell. This is called a graded potential. But in order to send long-distance signals all the way along an axon, you need a bigger change, one big enough to trigger those voltage-gated channels. That is an action potential. And your best bet for making that happen is to depolarize that resting neuron. I mean, cause a big enough change in its membrane potential that it'll trigger the voltage-gated channels to open. It all starts with your neurons sitting there at resting state. All of the ion channels are closed, and the inner voltage is resting at negative 70 millivolts. And then something happens. Some environmental stimulus occurs, say like a spider brushes up against a tiny hair on your knee, triggering those sodium channels to open, increasing the charge inside the membrane. Now, the stimulus and the resulting change have to be strong enough to cross a threshold for the true action potential to kick in, and that threshold is about negative 55 millivolts. Remember that number, because this is an all or nothing phenomenon. If the stimulus is too weak and the change doesn't hit that level, it's like a false alarm. The neuron just returns to its resting state. So kind of like Doc Brown hitting 1.21 gigawatts in the DeLorean, once it hits that threshold, not going to travel in time, but you are going to see some serious action potential. At that threshold, the voltage-gated sodium channels open, and there are tons of these. So all of the positive sodium ions rush in, making the cell massively depolarized. So much so that it actually goes positive, up to about positive 40 millivolts. This is action potential in action. It's really just a temporary reversal of a membrane potential, a brief depolarization caused by changes in currents. And unlike graded potentials, which are small and localized, an action potential kicks off a biological chain reaction which sends that electrical signal down the axon. Because each of your neurons has lots of voltage-gated sodium channels, so when a few in one area open, that local current is strong enough to change the voltage around them. And that triggers their neighbors, which triggers the voltage around them, so on down the line. As soon as all that's underway, the process of repolarization kicks in. This time the voltage-gated potassium ion channels open up, and those potassium ions flow out in an attempt to rebalance the charges. If anything, it goes too far at first, and the membrane briefly goes through hyperpolarization. Its voltage drops to negative 75 or so millivolts before all of the gates close and the sodium-potassium pumps take over and bring things back to their resting level. Now, when part of the axon is in the middle of all this and its ion channels are open, it can't respond to any other stimulus, no matter how strong. This is called the refractory period, and it's there to help prevent signals from traveling in both directions down the axon at once. So that is the surprisingly simple app that your nervous system uses to let you experience the world. And because the voltages in this process are always pretty much the same, the initial threshold around negative 55 millivolts and the peak depolarization at 40 millivolts, your neurons only communicate in a single monotone buzz. It doesn't matter if it's a spider on your knee or an elephant, a paper cut or a stab wound, the strength of that action potential is always the same. What does change is the frequency of the buzz. A weak stimulus tends to trigger less frequent action potentials, and that includes if the stimulus is coming from you, like your brain telling your muscles to perform some task. I need to do something delicate, like pick up an egg. The signal is low frequency. But a more intense signal, like trying to crush a can, increases the frequency of those action potentials to tell your muscles to contract harder, and the message turns into something that you can't ignore. Action potentials also vary by speed or conduction velocity. They're fastest in pathways that govern things like reflexes, for example, but they're slower in places like your glands, guts, and blood vessels. And the factor that affects a neuron's transmission speed the most is whether there's a myelin sheath on its axon. Axons coated in insulating myelin conduct impulses faster than non-myelinated ones, partly because instead of just triggering one channel at a time in a chain reaction, a current can effectively leap from one gap in the myelin to the next. These little gaps are the delightfully named nodes of Ranvier, and this kind of propagation is known as saltatory conduction, from the Latin word for leaping. But what happens when an action potential hits the end of its axon and is ready to do more than leap? Jump all the way to another neuron. That we'll find out next time. Today, we'll learn how your body is kind of like a big bag of batteries, and how ion channels in your neurons regulate this electrochemistry to create an action potential from resting state to depolarization to repolarization, and a brief bout of hyperpolarization. Thanks for watching, especially to all of our civil subscribers who make Crash Course possible for themselves and for everyone else.
Okay, that's enough. Enough for me. Hope it's enough for you, because I'm going home.